Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. Good evening. I'm Yvonne Stapp for Science for the Public, and I welcome you to the first of our four-part series of lectures on health and environment, featuring distinguished researchers who've worked for decades to raise public awareness of health environment issues. Leading off the series is an international expert on the effects of environmental pollutants on brain development, Dr. Philippe Grandjean. Dr. Grandjean is professor and chair of environmental medicine at the University of Southern Denmark and adjunct professor of environmental health at Harvard School of Public Health. He divides his time between Denmark and Cambridge so that we're able to have him here tonight. Dr. Grandjean received his MD and then his doctorate in medical science at the University of Copenhagen. He was a Fulbright scholar early in his career, and over the years he's accumulated many distinctions. He's a fellow of the American Association for the Advancement of Science and a board member of the distinguished International Academy Collegium Ramazzini. He's on the editorial board of 8 scientific journals and was also the founding editor of the journal Environmental Health. Perhaps more important to our audience, Dr. Grandjean's work led to a United Nations agreement to control mercury pollution. He received the Award for Excellence in Science and the Public Interest from 8 U.S. environmental organizations in 2004. Dr. Grandjean has spent decades studying the effects of environmental chemicals on brain development. He's conducted international studies that have set the standard for this type of research, and he has published hundreds of papers. Because Dr. Grandjean's work is so important, we have provided links to interviews and articles about his work on the Science for the Public webpage for this event, and we urge people to get informed by looking at that material. In addition, we urge people to read Dr. Grandjean's book, Only One Chance, which he'll be talking about tonight, which is very accessible and an indispensable account of environmental neurotoxins and the obstacles to testing them on the one hand and to regulating them on the other. We'll learn a lot about that tonight, I'm sure. And now we are very honored to present Dr. Philippe Grandjean.
I'm very pleased to be here. Thank you for coming. What I'm going to talk about today is a project that actually took me much longer to do than I normally like to do projects. I wrote a book. It started little by little, and overall it took about 10 years to do this, to get it finished. And obviously that book also relates to experiences that I've had for many more years than the 10 years. Anyway, it was published last year and has been well received. And it has some, I would say, implications that I'd like to share with you, because obviously there is medical science and epidemiology, toxicology, those various fields, but there are also social implications. And I think that we, as researchers at institutions like Harvard University and universities abroad, institutions have a responsibility to share the knowledge and help translate that into public policy as appropriate. So this organ we have between the ears is essentially an extremely complicated organ. The cells are packed in a unique way that happens in no other species in this world. So this brain operates on a minuscule amount of energy of about 20 watts. And you got to have one of those modern energy bulbs in order to be able to read just using 20 watts. That's all this organ requires in order to function. And if you take the best electronics people who can do programming and generate servers and try to make computers work together, that total computer capacity would require energy that corresponds to the average needs in Cambridge every day, just to do as well as your 20-watt brain is doing. This is how fabulous this biological structure we have is functioning. Now, it is, of course, a complication that as we look at our kids, the new generation, and how they grow, we try to give them the best chances to develop their central nervous system, to mature and to enjoy the benefits of having a good working brain. And we're putting in about $1 trillion per year in the educational system in this country. $1 trillion. It's something like 5 or 6% of the total national budget. But anyway, it's a sizable amount of money. Yet we know that schools fail. We know kids are being expelled from school. We know kids have problems of various kinds. We also know that psychiatric diagnoses like ADHD or autism spectrum disorder are increasing in frequency. We know kids get into problems with, some kids get into problems with the law, some kids get into problems of substance abuse, or there could be suicidal attempts and all sorts of problems. We know they happen. And the problem is, how come if we spend so much on the educational system and parents do as well as they can out of love for their kids, how come we still see these problems and they're growing in magnitude? Well, perhaps we need to look back and look for causes in the children's background, the development of the brain. Now, what we know from modern science is that we have made some very serious mistakes in the past. We have made assumptions that I today would characterize as naive and dangerous. Number one, we assume that when a woman is pregnant and she's nurturing the fetus, we assumed, when I went to medical school many moons ago, I was taught that the placenta was able to filter so that the oxygen and the nutrients would get through to the fetus, and all the waste products and toxic substances would never make it to the fetus. That's not true. Environmental chemicals, industrial products and chemicals pass the placenta with very little delay. Second, we also assume for many years that, well, children are just little adults. When we take the body weight into consideration, they are as sensitive to poisons as other and older people. Wrong. We thought traditionally, just because in the hospitals we usually see a kid has, let's say, pneumonia, and after the infection has worn off, the child is, just as fine as before. We thought that's the same thing with chemical exposures, that they are reversible. So that the exposure happens, and when it has been eliminated, the child is fine again. Not true. We also encountered some resistance in that chemical products are, of course, useful to society. And the dictum in the old days, and I'll return to this, the dictum was, all right, but if you think that these chemicals are toxic, you've got to prove it. so that the assumption was that everything is innocuous and inert, unless proven otherwise. And that's really a dangerous assumption. And the final one is that testing chemicals for their toxic effects is too expensive. In this country, we have legislation. It's called the Toxic Substances Control Act. It was enacted in the late 1970s. It doesn't even require that we test new chemicals for toxic effects before they're marketed. That's what you have to do in the European Union, the so-called REACH legislation, but not in this country yet. So these assumptions have endangered brain development in the young generation and in the generations to come. And I'll tell you why. We have discovered in neuroscience and biomedicine that brain development is a uniquely complicated affair. What happens is that the cells start as a little strip that expands in size, and then that little bulb has to expand greatly. And for every second at the peak, 200 new nerve cells are created. Now, those nerve cells have to differentiate. They have to become, there are different types of cells in the brain. They have to separate into different populations. So they do that. They master that as well. And then you can imagine if this starts as a little bulb and the whole thing has to include a cortex, then those cells have to migrate from their initial birthplace and figure out, wait a minute, I gotta go right here. Okay, all right, left, straight. So they have, and they actually sniff their way. They sense chemical signals, and then they work their way to their final position, and they arrange themselves in columns in certain parts of the brain so that they are in an ideal position to start communicating. Now, those brain cells have to figure out exactly the way to do this. And if that doesn't happen, you're in great trouble. So they also have to send out extensions to communicate. You know, they have to communicate all the way down to your big toe and your pinky finger. And if you take those extensions called axons, you put them lengthwise from one person, can you figure that out, that experiment, you will reach 4 times around the globe. That's the total length of these nerve extensions that you have in your body, 4 times around the globe. So there are little contact points where each nerve cell will have maybe 1,000 contact points called synapses. And just after birth, the generation of synapses happens at a speed of about 1,000 per second. But then, of course, some of those contacts are not very useful. And you know, from an infant sort of trying to move and learning to control the body and perhaps utter some words or sounds, all of that takes coordination. And the synapses that are useful are kept. And then those that turn out not to be useful, they are weeded away. So that process of weeding the synapses is also very important. Now, all of this is necessary for your brain to turn out to be that extremely complex and uniquely functional organ, which, you know, makes you who you are and distinguishes you from all other species in this world. The point is that these processes, because they are so complex, they're also uniquely sensitive. If something goes wrong, just a tiny little bit, the consequences can be serious. So we first learned that in the 1950s. Here's a picture of a mother, a Japanese mother from Minamata. She was eating contaminated fish during her pregnancy. And you see she's bathing her child, and the child has severe spastic paresis and mental retardation. This child was born with congenital mercury poisoning. The mother had been eating contaminated fish, but she was completely unscathed. So the mercury passed through her body without causing damage. But when it moved through the placenta into the fetal blood circulation and the fetal brain, then it caused this severe damage. So the observation from one of the Japanese doctors who tended to these patients was that the mother was healthy, and then the child, just after birth, it's very hard to make any diagnosis in newborn because there are so few tests that you can use, but a few months after birth, it was very clear that that child was abnormal. That was how Minamata disease was discovered, and that was what last year led to the Minamata Convention of the United Nations. I'm going to show you another slide from Minamata because there were hundreds of people who died from mercury poisoning. And what the pathologists did was to conduct an autopsy, and then they examined the brain tissue. And so there were adults who died with, and the lesions were located here in the cerebellum. This is a visual cortex. And people had tunnel vision as an indication of the damage here. They had some damage in the motor cortex. They had difficulty with coordination of movements. Children who died having been poisoned as children after birth, they had more widespread damage, and kids who died from prenatal exposure to methylmercury, you can see that they had widespread. I've seen pictures in the microscope. I mean, I've seen the slides from these kids. The whole architecture of the brain is completely disorganized, and which means that the brain cells didn't make it to their right positions. And that's, of course, why those kids get mental retardation and spastic paresis. Now, the important thing is that this happened at much lower doses than the doses required for later on in life or as adults, where the damage was much more limited. Given that experience from Minamata, we of course wanted to know, is there a problem with the mercury that we eat in fish and you know, tuna or saltfish or whatever it is? So we started doing studies in the Faroe Islands, and we've also worked in Greenland, and there have been projects here in the United States. And I wanted to show you just one picture because there are, I could have shown you many tables, but this picture I think tells you a whole lot of what we have found. This is a functional MR scan. And what happens with this technique is that the brain area that is currently being used will light up with orange here. And this is a teenager we've asked to do this with the fingers, so finger tapping. And because it's the left hand, you know, the nerves cross, then it's the right hand motor cortex that should light up, and it does. Exactly. So that's completely according to the theory. Now, we also did that with kids who had a prenatal exposure, a high exposure to PCBs and methylmercury. And look, they have to use both sides of the brain in order to do this with the left hand. We've looked at some other types of stimuli, and every time we looked, those kids with This is an average picture. Those kids with a prenatal exposure, they had to use a much larger brain area in order to do something very simple, as an indication that the nerve cells did not have the right connections, and therefore they were asking for help from nearby areas. This is, of course, very abnormal, and this is clearly also associated with decreased functioning. I wanted to show you something a little bit closer to home, because there are data from Massachusetts that you can actually access. This is the MCAS math score from the fourth grade. And what Jessica Reyes did here was to access the averages. She couldn't get individual data, but she got the averages from the 351 school districts. And then she matched that math score to the average concentration of lead in the blood. And you know, you have to get the blood lead concentration measured at preschool age. And she took the average for each child before age 6, having excluded the highest value. So, and you can see there's some sort of association here. These are kids born in 1992. Here, these are the records for the kids born in 1999. It looks like if you have less lead in the blood, you're also doing better. Same thing here, but here's a shift. This is like a little bit further up to the left, and it becomes really clear. I'm now putting both of them together on one slide. So this is the 1992 kids. They're a little bit lower in math score, and they're higher in the blood leads. And look, these are the younger kids who have been exposed to less lead, and they do better. Well, I'm not saying this is a proof that lead necessarily does that, but this has been replicated also in Rhode Island. It's been replicated in Minnesota, in Wisconsin, I think Michigan. Various states, they have the same types of data, and they've been able to show that there is an association. The higher the blood lead concentration, the worse off is a kid. Now, this is, of course, also associated with poverty and ethnic background. And the interesting thing is that lead exposure is also associated with poverty and ethnic background. And it turns out that lead can explain a good part of what we thought was due to ethnic background and poverty. It's not really poverty as such. Well, it does something by itself, but some of it is due to lead exposure, because poor people live in they have a greater risk of living in a dilapidated house with lead paint, with water pipes made of lead, other sources of lead exposure. So this is it's a social problem in this country. So CDC a couple of years ago decreased the current exposure limit down to 50 micrograms per liter, or 5 micrograms per deciliter, as they say. But anyway, here's the limit. According to CDC, that is a safe limit of lead exposure. I'm sorry, this is bullshit. It is not safe. And I'm sorry for using words like that, but the World Health Organization has looked at all of the literature that has been written about lead, and they concluded a couple of years ago that there is no safe limit for lead exposure. Lead is so toxic that even the lowest concentrations here are still affecting brain development. So we got to do better than what CDC is urging us to do. Of course, it would be great if all of the kids could be below this concentration, but I would prefer they were much below, not just below. I've now talked about some of the you might say, the usual suspects, namely lead and methylmercury, and arsenic is also one of the poisons that we've known for a long time. Just a week ago, a paper was published from Maine, because in Maine, about 20% of the households have a high arsenic concentration in the drinking water. And the study that came out, it was done by colleagues from Columbia University. They showed that kids who had an increased arsenic exposure from drinking water, they also had a poorer IQ. They had lost an average of like 5 or 6 IQ points. Now, high arsenic concentrations in the drinking water is common in something like 20% of the drinking water sources in Maine. So this too, like lead, like mercury, is a common problem. Ethanol is on the list. It's not really an industrial chemical. It's of course related to fetal alcohol syndrome. The literature that I know of suggests that if you're pregnant, simply abstain from alcohol, because typically it's very much like lead and arsenic and these other chemicals we know about. Even small doses are toxic to the brain development. Toluene, we don't know as much about it. It's a solvent, but there was an unfortunate period in the 1980s and 1970s where toluene was used for sniffing. And pregnant women in some parts of the United States used that for a period, and when they used it during pregnancy, the consequences were just dramatic. Those children suffered serious brain damage. So those are the ones that we identified along with PCB. That's a contaminant also in seafood. You may have heard about New Bedford, where there's a PCB contamination of the harbor. You probably also know the Hudson River, a very serious PCB contamination. So we published that in 2006. And then we were eager to follow, are we going to find more chemicals that cause brain drain during the following years? And the article just came out a month ago that we were able, in the most recent years, to find another 6 chemicals. And 2 of them are inorganic, namely fluoride. And basically, the fluoride is based on research carried out in China. And it's because fluoride occurs as a contaminant in drinking water. It's a natural contamination, which also happens in this country. And because of the circumstances where farm families essentially stay all their lives in the same location and they have the same source of water, then it's possible to carry out studies to find out if children who have been exposed prenatally and as children to high fluoride levels in drinking water, if that's related to deficits in brain function. And the average of those studies in China, some of the kids had very high exposure, some of them similar to high levels that occur in this country. The loss in IQ points was about 7 IQ points on average. So I mean, this is a sizable loss in IQ. Manganese has been studied in Canada and Mexico in particular, and from industrial pollution primarily. And then we have tetrachloroethylene. The best studies are from Cape Cod, where there was tetrachloroethylene contamination of the drinking water. Chlorpyrifos is a common pesticide that occurs as a food contaminant. DDT and DDE it's a very stable, very persistent and accumulative chemical, and we're all exposed. We all have DDT or the metabolite DDE. We all have that circulating in our blood. And this has also been associated with brain deficits in children. These are the brominated flame retardants. They're now banned, but they still occur in like television sets or other electrical equipment manufactured before something like 2006. They've been phased out, and there are other other brominated flame retardants on the market. Unfortunately, those here that have been phased out that we now know a lot about, we know that they're toxic to brain development. So, and they still occur. They are, and we can always find them in house dust from old equipment. But the new flame retardants, we know much less about. And therefore, we don't know if it's safe. And so we're coming back to the problem of whether or not we should test such chemicals for those kinds of properties. Now, having talked a little bit about the historical cases and what we've learned recently, I want to share with you the overview of how we have learned new information in this field. What happens is that with time, we discover that more and more kids are affected by these chemicals. And obviously that is, we find that there are more kids affected, but at a lower dose, because we'll learn that the damage happens even though we thought the level was safe. Just like CDC was thinking that 50 micrograms per liter in the blood is safe. No. The new research shows very clearly it is not safe. So we learned in the beginning that the chemicals were toxic to the brain, for example, in occupational health. The workers were exposed and they got various kinds of neurological symptoms. There were some poisoning incidents where perhaps even children got affected. And later on, studies were done, like Herbert Needleman did here in the late 1970s in Cambridge. He studied lead here, right here, and discovered that more kids were affected at lower doses. And what we're seeing today is what I call the silent pandemic. It is worldwide. It happens, it affects an enormous number of children. It happens at fairly low doses, and it's silent because only a small minority of those kids have a diagnosis. Some of them will have spastic paresis because they just went over the edge and then they got really sick. Some of them have ADHD. Some of them have other diagnoses. Some of them have dyslexia. Some of them just function poorly and they don't really have a diagnosis. Most of the kids are within the normal range. They just don't have optimal functioning. So that's why we call it the silent pandemic. And it's particularly nasty and insidious because you can't see it. You can't diagnose this because it's only apparent maybe at school age, and to identify the exposures way back during brain development that caused that is very difficult, which also means that there's a complication in doing research in this area. So the chemical universe, I don't know what your number is, but it's between 50 and 100,000 chemicals. And basically, we know only a little bit about that universe. Something like 1,000 chemicals we can see are toxic to brain tissue in laboratory experiments. And we know that at least 200 industrial chemicals have caused poisoning in adults with symptoms from the nervous system, which means that those chemicals can pass into the brain and cause toxicity from the brain cells. So 200 chemicals here. About half of them are pesticides. That's interesting, I think. Half of them are pesticides, and obviously pesticides are manufactured to be toxic. I mean, that's why they're generated. And unfortunately, if they are targeted, if they're targeting insects, insect pests, then the best way to do that is to target the nervous system. And I don't want to embarrass you, but quite frankly, the biochemistry you have in here is not all that different from what a cockroach has. I'm sorry, but it's a matter of fact, which means that the chemicals that are affecting those pests may also affect your brains, or in particular, the fetal brain that you're nurturing. So the number of compounds I showed you before is now a total of 12. So you can see this is really a tip of an iceberg. And basically, as you can understand, we're not really doing enough to regulate lead. We're not doing enough to regulate mercury or arsenic. So we're not even taking a proper crack at solving these problems up here. But what about all of this here? It is a big issue, and it's one that we need to address. So here's the situation, and I wanted to show you some of the risk factors, and here they come, like solvents and pesticides and stresses and fluoride and tobacco smoke. And so these are the risk factors that we know today. And there are a few more that I could have mentioned also. But to carry out research in this area is extremely complicated. We can't do experiments and invite pregnant women inside and say, would you mind taking a little mercury because we'd like to do this experiment? There's no way. We can only observe because some people happen to have a higher exposure to, one substance or another. And then sometimes they're also exposed to, let's say, tobacco smoke or crack or alcohol or whatever it is. And so we have to adjust for that and try to see through the fog and figure out what's really going on. And you can only test the kids when they are at a particular age and they are capable of following instructions and know that they have to do their best to perform well on the test. And that means that these kinds of studies, they take many years to do. And there are also weaknesses, and then there are, we call them confounders, when there are other factors that need to be taken into account. So what do we then do in practice? Well, I'm showing you this picture of a gentleman whose brain is on a scale. And the fact of the matter is that the type of animal studies that we actually do for testing chemicals involves measurement of the brain weight when rat pups are born. So they take out the brain, put it on a scale, and if the brain weight has decreased by more than 5%, they say, this compound is probably toxic to brain development. Excuse me. Putting somebody's brain on a scale is not going to work. I've looked at the information that's available on human brain weights, and I've found out that one of the smartest people who ever lived, his name was Einstein. He, for some reason, had his brain removed when he died, and somebody put that brain on a scale. And you know what? It was subnormal. His brain was small. And to believe that we can just put brains on a scale and figure out if something has gone on and damaged your brain development, bordering to the naive. I mean, this is like, it's almost criminal. But this is the way we are testing chemicals today. There is an OECD protocol, which we're beginning to use, but only occasionally, and the EU is currently debating whether to make this mandatory for chemicals in the future. It'll take a few years. We have the methods. We can even test chemicals in Petri dishes. There are methods to examine stem cells, nervous system stem cells in Petri dishes. We have the methods. We can do it. But there's a cultural problem here. Several years ago, there was a conference organized by the International Life Science Institute with the United States Environmental Protection Agency and several others. And they discussed, are children really more, and the fetus, more vulnerable than people are on average? And the conclusion from that meeting, and that is a conclusion that is, I would say, almost universally accepted. The conclusion is maybe, because differences must be studied and evaluated on a case-by-case basis. That's what the conclusion was. So we need to document this in every single case. That's a line of thinking. I believe this is irresponsible. It is a conclusion that must be seen in perspective, and the perspective is the next slide. Because here's the chemical universe, and that's just what we call the high production volume chemicals. There's almost 3,000 of them. The great majority of those chemicals have not been tested for anything. And if we insist that the sensitivity of the nervous system during development must be proven for each and every one of those chemicals, that's the same thing as saying, let's not do anything about it for the next multiple decades. This is, and waiting for decades means, all right, let's experiment on another generation of kids while we're watching what's going on. And so I think this kind of conclusion needs to be reached based on, I'd say, an open discussion, and not just on a matter like where you look at the strength of the evidence and how much has been proven beyond doubt. We have a social responsibility here. Let me illustrate this bit by this curve here that illustrates the distribution of IQ. And I focus on IQ because it's a common term, but chemicals that affect brain development affect not only the IQ, they also affect behavior, and they can cause psychiatric disease. They can cause an increased risk of delinquency and substance abuse and all sorts of other problems. But let's just focus on IQ, and you can see that there's a small proportion that would be classified as mentally retarded because they have an IQ below 2 standard deviations, below 70. And then there are the very smart people who attend meetings in Science for the Public. But then, if there's a shift, and here we're shifting this downwards by 5 points, you will see that this proportion here is increasing substantially, and there's going to be problems for meetings with Yvonne and Science for the Public in the future, because there's going to be fewer smart people that are going to help create innovations in the modern knowledge society or information society. So the consequences of this shift, even though you could say all of those kids here, they are within normal ranges, they're not sick. There may be some here that are classified as sick, they get a diagnosis, but most of the problem is here. Kids who have suboptimal functioning. I should tell an anecdote here at this point, because Herb Needleman, whose name I mentioned before, who studied kids here in Cambridge who were exposed to lead, and found out that the higher the lead exposure, the poorer their school performance and pure cognitive functioning. So he gave a talk at Harvard University, and a very impressive talk, I understand. I wasn't present. But after the talk, a colleague wanted to ask a question, and this colleague said, "But Dr. Needleman, you've shown that lead exposure causes these significant, but only slight changes, in fact, because all the kids were healthy, and you know, just losing a few IQ points. Is that really something that's going to change the way we run society?" And then Needleman said, "Dear colleague, I know many people here at Harvard University who would give a fortune for just one extra IQ point." So, and that's exactly the point. Wouldn't we all want to have optimal brain functioning, and wouldn't we want our kids to have that? To have the greatest advantage and pleasure out of a well-functioning brain. I think it's crucial. You can donate one kidney if you want to a relative who needs a kidney transplantation. You can have a perfect life with just less than half of your liver function. You have lots of reserve. You could say you have lots of reserve in your brain, but that reserve is exactly what makes life particularly fun. And that's what we need to protect, because it's also important to society. So in the large scale, IQ points have been used by social scientists to identify what are the costs to society if we lose them. And this has been done in regard to psychiatric and medical diagnoses like these, autism here, ADHD, and you can see that the number of IQ points lost per year, these are very substantial. This is David Bellinger from Children's Hospital here in Boston who's done these calculations. And when you look at the chemical exposures, you can see that those numbers here are the same order of magnitude. And if you add, I mean, this is just for some pesticides, mercury and lead. If you add arsenic, perhaps even fluoride, manganese, solvents, tetrachloroethylene, etc., the total number of IQ points lost becomes quite substantial. Social scientists have calculated that your lifetime income, and this is something I've been talking about, the pleasure of having a well-functioning brain, but it's also important to society because you will be more economically active and you will generate more taxable income the smarter you are on average. So social scientists have calculated that one IQ point in terms of lifetime income and then discounted to the value in terms of money in the hand today. That value is about $15,000 per IQ point. Well, I don't think I will dispense of any of my IQ points for $15,000 a piece, but anyway, that's what social scientists have done. And in regard to lead, if you calculate, these are for 0 to 5-year-old kids. And in regard to lead, you can calculate that the annual expense is $50 billion at the current exposure levels, including the levels that you saw here in Massachusetts. So $50 billion, compare that to the $1 trillion that we're using for the educational system. Now, if you add a few more of these environmental chemicals, we're moving upwards. And it looks like perhaps some of that money we are using elsewhere in society might be very well placed in trying to prevent these exposures from happening. At least this is very much in accordance with the economist James Heckman, who is a Nobel laureate and who has been promoting educational programs for children, where he says that the benefit, the return to, he's an economist again, so he's talking about the return to investment in human capital, and it's something like $8 or $10 or more per dollar invested if you use the money for preschool programs or even earlier programs to help children develop, stimulate their brains, mature. It's an extremely good investment. My question is that prevention of chemical brain drain, wouldn't that also be a uniquely attractive investment? The problem is, and that's why there's a question mark, we don't know exactly what it's going to cost to take away the lead from old housing, the arsenic from the drinking water, etc. We don't know exactly what it's going to cost. But it is very clear that chemical brain drain causes a lot of damage to the next generation's brain development. So, having said all of this, what can we do? What can we do to inspire better public policy now that we know so much about the vulnerable brain development and the types of damage that can happen to brains due to industrial chemicals? Well, there are three problems with the kinds of science that we have access to, that's available to us. And the crucial problem is that we just don't have the systematic data. I told you we're now up to 12 chemicals that we know pretty well that they cause chemical brain drain in children. We're just beginning to control those chemicals. But we don't know much about the rest of the, let's say, 100,000 chemicals out there. We don't. So it's a really big problem that we don't have systematic data. And there's no intention from, let's say, the US EPA or FDA or CDC to fund this kind of an activity. Maybe they can do it in Europe, but the research program that President Obama presented a couple of months ago, the BRAIN program, the intention of that program is to use better electronics models to model brain functioning. So they're interested in figuring out how the brain works, and they think that this is going to have an important effect on commerce and stimulate production in this country. I'm not sure I understand that, and I'm not sure that it's realistic, but I am sure it is realistic that if we did more to identify chemicals that damage brain development, we could generate kids with brains that are functioning much better and with less of these problems that I've mentioned. The other problem that's common is what I call a bias toward no effect, a bias toward the null. And I don't know if you saw that article in The Economist last fall, where The Economist claimed that, journals like New England Journal of Medicine, they claim that they find something very important, and a few months later it turns out to be wrong. So science is often wrong, and therefore, forget about it. Science is not useful for anything. That's what The Economist essentially said. They even had a vote where the readers could, hit a yes or no button, whether they believed in science. Come on, I mean, how populistic can you be? The problem is really that people like myself, when we conduct studies, there are so many reasons that we may miss that mercury is toxic to brain development. It could be that we were looking at too few children. Maybe we looked at them when they were different ages. We couldn't get them to come at the right time. Maybe we had some problems that they were examined by more than one psychologist, and then there were all sorts of noise going on. All of these things mean that we have a bias toward finding no effect. So the research that we have in front of us is really underestimating the true magnitude of the problem. And then there's a problem that I must admit that it's very embarrassing. I've been reading my own publications, those from a while ago, and I tend to write, kind words like maybe and perhaps and under certain circumstances. And within this particular study, this needs to be verified, and replication would be nice. You know, this kind of thing that researchers do because they sort of negotiate their way to the conclusions, and they tend to be modest and humble. At least they would like to appear modest or humble. And this what I call hedged language is oftentimes misunderstood. And decision makers would say, no, but these scientists are writing maybe and perhaps, and they say it needs to be replicated. Come on, how much replication do you want? I mean, if the kids really show serious damage to brain development, and we know that this is associated with this particular exposure, how many kids in addition do you want to see before you believe this? I mean, you gotta see a perspective here. But the point is that, that when we write these conclusions, we are polite and modest. And it needs to be interpreted the right way. All right, so here comes this boy. And so the conclusion from all of what I've said today, from biomedicine, from neuroscience, is that we have finally reached a conclusion that there is only one chance to develop a brain. And that period that starts prenatally, especially the third trimester of the last 3 months of pregnancy, it's a very vulnerable period. You only get that one chance. You can't go back and then reconstruct it. You can't. And our brains are so complicated, and the price we pay for that is that the development is uniquely sensitive to toxic chemicals. It's the early development. With time, the brain gets less and less sensitive. Of course, the fully developed brain is not there until we are like 18 or 20, but basically I'll say up to the teenage, then there's a substantial amount of susceptibility, but the main susceptibility is late pregnancy and the first couple of years after birth. And we have been examining kids up to age 22, and we continue finding the same thing. We're finding that the damage that was present at age 7, it's also there at age 14. We find it again at age 22. It doesn't go away. And of course, if your brain cells are not in the right places and they haven't been able to make the right connections, you're not going to have a second chance to build that. So of course, this is why it's permanent. So the goal, I would say, is that we need the full integrity of the brain, because that is what allows optimal brain functioning. And we need it because the brain determines our behavior, our IQ, even endocrine functions, and it determines who we are. So here's the conclusion. We have a brain that we're trying everything we can to nurture and stimulate and help grow, but there's also a drain happening here, what I call the chemical brain drain. And I think that we need to interpret our current scientific understanding of brain development to try to turn off that faucet. Thank you.